Ever catch yourself, lying awake at night, thinking about the big stuff, like what does it even mean to be alive? Or how do these crazy complex systems we see in nature, how do they actually work? It's kind of like, what's the key to truly understanding how the world ticks? Well, if that sounds familiar, then this deep dive is right up your alley. We're going to be looking at systems and synthetic biology.
Two fields that are, really grappling with those big questions, and not just theoretically. I mean, they're actually going in there, doing experiments, designing new biological systems. It's really pushing the boundaries.
And for anyone out there who loves to dig deep, the philosophical types who can't resist a good thought experiment, well, these fields offer a really unique perspective. We're going to be using the Stanford Encyclopedia of Philosophy's entry on the philosophy of systems and synthetic biology.
A great resource. Really comprehensive overview of, the philosophical implications of this whole area.
So think of this deep dive as us, kind of like taking that deep philosophical analysis and pulling out the nuggets that are going to really resonate with, that philosophical mind. What we really want to get at is how these two fields, systems and synthetic biology, how they tackle those classic philosophical questions. Like, what does it mean to truly understand life? How do the parts of a system relate to the whole? What even makes for a good explanation in biology? And what does it even mean to talk about design when it comes to living things? Heady stuff.
It really is.
Yeah.
So to start us off, maybe we should define our terms a bit. Systems biology, synthetic biology, they sound related for sure, but what's the core difference between the two?
Right. So systems biology, it's all about understanding, right? Like trying to really grasp how living systems work as a whole, not just breaking them down into their individual parts like genes and proteins, but seeing how everything interacts, the dynamics of the whole system. It's kind of like instead of looking at individual instruments, you're trying to understand the whole orchestra, how all the musicians work together to create the music.
Makes sense. And then where does synthetic biology fit into that?
So synthetic biology takes that understanding and it's like, okay, can we actually build something new using what we've learned? Can we design and build biological systems? Kind of like an engineer designs, a circuit or a machine, but using biological parts.
So one's about analysis, the other's about creation, but they're definitely feeding into each other. Mm. And both rely on this really interdisciplinary approach. They're drawing from biology, obviously, but also math, computer science, physics, chemistry, engineering. It's a real melting pot.
Now, what was it about the way we were doing things before, like traditional molecular biology? What was it about that made scientists think, hold on, we need a whole new way of looking at this? What were the limitations they were bumping up against?
Well, molecular biology has been no doubt incredibly successful, right? It's given us this detailed understanding of how life works at the molecular level. But it's kind of in the name, right? It focuses on the molecules. And often things are studied in isolation. But living systems, they're anything but isolated. They're these massive interconnected networks of interactions. So you could study, say, a single protein in a test tube, but that might not tell you how it actually behaves within the complex environment of a living cell, with all the other molecules it's interacting with.
It's like trying to understand a sentence by just looking at the individual words. You lose all the grammar, the context, the meaning.
Exactly. And systems biology came about because scientists realized that to really get a handle on life, you need theoretical frameworks, computational tools, things that can deal with this interconnectedness. It's about looking at the dynamics, the organization of these vast networks. How do they respond to change? How do they regulate themselves? All these really complex questions.
Okay, that makes sense. Like, we can know all the ingredients in a cake, but if we don't know how they work together, how the baking process works, we're not going to end up with the cake, right? So then what sparked this whole synthetic biology thing? Why start thinking about building biological systems from the ground up?
A few things, really. I mean, genomics, it was advancing like crazy. Mm-hmm. We were suddenly getting this incredible understanding of the blueprint of life. But there was also this feeling that, we could do more than just tweak existing biological systems, which was kind of the approach with traditional genetic engineering. It's like, okay, we have the parts list, but can we write our own instruction manual? Can we design biological systems with specific functions in mind?
Kind of like instead of just tinkering with existing machines, you're designing entirely new ones from scratch.
Right. And there's the potential for, all sorts of practical applications, new medicine, sustainable energy, environmental sensors, who knows what else. But there's also this deeper, almost philosophical drive. Like, can we really understand life if we can actually build it?
Building life from scratch. Man, that sounds like something straight out of a philosophy seminar. What are the actual research avenues they're pursuing in synthetic biology?
The source mentions a few key areas. One is using these standardized biological parts called biobricks to build DNA-based devices. It's like having a set of Lego blocks, but instead of plastic, it's DNA. You can mix and match them to create new functions within a cell. Pretty wild stuff.
Wow, biological Legos. That's an image.
And then there's genome-driven cell engineering. Scientists are working with minimal genomes, which is like the bare minimum set of genes that are thought to be essential for life, trying to figure out what's absolutely necessary, you know. And then, of course, there's protocell creation. That's the attempt to actually build artificial cells from non-living materials. That really gets to the heart of the what is life question.
Yeah, talk about a heady question. Minimal genomes, that's basically asking, what's the bare minimum you need for something to be considered alive, right? To be self-sustaining.
Yeah, and researchers have actually managed to create a synthetic minimal bacterial genome. They inserted it into a cell and boom, you got a functional self-replicating organism. It's pretty mind-blowing when you think about it.
So all this brings us to this concept of design, right? We usually talk about biological systems being shaped by evolution, natural selection, all that. But systems in synthetic biology, with their, focus on engineering, that kind of changes how we think about design in biology, doesn't it?
It really does. I mean, bringing in these engineering concepts, it forces us to reconsider what design even means in this context. Sometimes they use what they call a thin notion of design. It's basically just focusing on the relationship between a biological structure and its function. So instead of asking, why did this function evolve? They're asking, how does this particular arrangement of molecules actually achieve this specific outcome?
So shifting the focus from the historical why to the more immediate how.
Yeah, in a way. But then that raises a really important question. Can this thin notion of design really capture the full complexity of biological systems? Like, what about their autonomy, their ability to act independently, to evolve and adapt, and that sense of, intrinsic purpose that we often associate with living things? It's hard to pin down scientifically, but it's definitely there. And then there's their sensitivity to context, and this really interesting concept called degeneracy.
Degeneracy. That's a new one. I'm guessing it's not as negative as it sounds.
No, not at all. Imagine you have two different sets of components in a biological system, let's say two different combinations of genes and proteins, and they can both achieve the exact same function. That's degeneracy. It's like having multiple routes to get to the same destination. And this makes biological systems really robust. If one pathway gets disrupted, there might be another one that can take over.
So it's like a built-in backup system. Pretty clever. So how do researchers in these fields actually go about designing stuff? Is it just like drop a blueprint and build it?
Well, the source talks about forward and reverse engineering. Reverse engineering is like taking apart a clock to figure out how it works. You start with an existing biological system, you study its structure, its function, and you try to deduce the underlying design principles. Like the search for those network motifs we talked about earlier, those recurring patterns in biological networks. That's a form of reverse engineering, trying to figure out what those patterns tell us about the system's capabilities.
And forward engineering is the more intuitive one, right? Like you have a goal in mind and you try to build a system to achieve it.
Exactly. You define the function you want, and then you try to figure out what biological parts you need, how to put them together to make it work. And this brings up a really interesting philosophical point about what counts as an explanation. In systems and synthetic biology, sometimes just demonstrating a plausible mechanism for how something could work, even if you haven't proven that's how it works in nature, can be a really important step. It opens up the possibilities.
So even a theoretical model, as long as it shows a possible way for something to happen, that can be valuable in itself.
Absolutely. It can reveal fundamental principles, guide new experiments, inspire new designs. This gets at the idea of design principles in biology. These are recurring patterns, strategies that seem to underlie lots of different biological functions, like feedback loops for regulation, or those network motifs we keep coming back to, scale-free networks, bow tie networks, feed-forward loops. Researchers are really interested in these common architectural elements, thinking they might be like the fundamental building blocks of biological function.
You mentioned those network motifs a few times. Can you give us an example? Like, what would one of those actually look like?
Okay, take a feed-forward loop. It's a specific arrangement of three interacting components. And in gene regulation, it can create a delay in the expression of a gene. It's like a logic gate or a filter that only responds to sustained signals. The idea is that these recurring patterns might be responsible for generalizable functional behaviors across different biological contexts. And there's some pretty compelling evidence, especially in bacteria, that supports this link between specific motifs and specific functional outcomes.
Okay, but can we really understand how something works just by looking at these like network diagrams? What about the actual molecules involved, the environment they're operating in?
That's a big question. There's this debate, right? Some argue that the topology, the wiring diagram of these networks is crucial. Like that can tell you a lot about how the system's going to behave. Others say, hold on, you can't ignore the specifics, the particular proteins, RNA molecules, all that stuff. And the context, the cellular environment, that matters too. You could have the same network structure, but it might behave very differently depending on the specific components and the conditions.
So it's like saying the same computer program could run differently on different hardware.
Right. And this connects to this philosophical idea of multiple realizability. It's the idea that you can achieve the same function using different physical substrates. Some argue that these network motifs, they provide real-world biological examples of this principle, and understanding them could give us like design heuristics for synthetic biology. If we know a certain motif achieves a certain function, maybe we can build a similar motif using different biological parts.
Makes sense. Now, what about the overall organization of these systems? Are they neatly compartmentalized, like a machine, modular,
Right.
or is it all just one big interconnected mess?
Another big question, modularity, the idea that you have these like self-contained functional units, it's very appealing from an engineering perspective, would make design a lot easier. But many argue that biological networks are way more interconnected. The interactions between different parts, they're crucial for how the whole thing works, for its robustness. Systems biology is really trying to get a handle on this interplay between modularity and holism.
We talked earlier about this ideal of rational design in engineering. What's it actually like doing research in synthetic biology? Is it always a straight line from blueprint to working system?
It's definitely messier than that. This source talks about this idea of reverse and forward tinkering. It's more like an iterative process. Trial and error, exploring different combinations, seeing what works. It's a bit more like biological kludging, finding solutions that work, even if they're not the most elegant.
So less like an architect with a perfect plan, more like an inventor in their workshop, just, trying things out.
Yeah, there's definitely that element. And the source even talks about this reflexive double-bind engineering. Basically, the very act of trying to engineer biological systems, it often reveals these fundamental differences between biology and our traditional engineered systems. They call these negative analogies.
Can you give an example of one of these negative analogies?
So one example is the role of noise. In engineering, noise, like random fluctuations, is usually something you want to get rid of, right? To achieve precise control. But in biology, noise at the molecular level can actually be functional. There's this synthetic genetic circuit called the repressilator. It was designed to oscillate, but it didn't behave exactly as predicted by the mathematical model. And this led to the realization that, well, maybe this inherent noise in gene expression, maybe that's not a bug, but a feature of living cells.
Wow. So trying to build something precise actually showed them how imprecise these natural systems are and that imprecision might even be important.
Exactly. And it shows the limitations of just taking engineering principles and applying them to biology. It's a different beast. And it raises the question of whether our design strategies need to account for these inherent biological properties like noise, context dependence, all that.
This makes me think about, the ultimate goal of some of this work, especially that protocell stuff. They're literally trying to build life from scratch. Yeah. How does that, square with the whole philosophical question of what is life?
It's engaging with that question head on. Researchers are trying to figure out, what's the bare minimum you need for something to be considered alive? They're experimenting with lipid compartments, metabolic processes, catalytic RNA molecules called ribozymes, basically trying to build simple artificial cells and see what happens.
And are they getting any closer to a universally agreed upon definition of life?
Not really. The source actually emphasizes that we still don't have a universally accepted definition. And there's even a debate about whether finding one is even the right goal. Some argue that it might be more useful to have operational definitions specific to certain contexts. Like if you're building models or searching for alien life, you might need different definitions.
So the very act of trying to build life is forcing us to confront the question of what life even is.
Exactly.
Yeah.
And it ties into other philosophical discussions. Like, does the definition of life have to include evolution? And how do we even define evolution? And can we really lump all the different forms of life we see on Earth under one definition? It's a big philosophical can of worms. The source even mentions how ideas from classical philosophers like Aristotle and Kant and more contemporary thinkers who focus on systems theory, how all that is relevant to how we think about causation and organization in living systems.
Okay, so we've talked about how these fields are trying to understand and even build life. Let's circle back to those big philosophical questions we started with. How does all this relate to the whole reductionism versus holism debate?
That's a core issue here. People who champion systems biology, they often see their approach as an alternative to what they call modular reductionism. It's this idea, often associated with traditional molecular biology, that you can understand a system by just understanding its individual parts in isolation. Systems biologists argue that the interactions, the overall organization, that's crucial for understanding how the system behaves.
So it's like a modern biological argument for the idea that the whole is more than the sum of its parts.
Exactly. Systems biology gives us a framework for understanding what that more actually is, those emergent properties that arise from the complex interactions within a system. And lots of people argue against reducing higher-level phenomena to lower-level explanations. Like when you're modeling something complex, like the human heart, you can't just look at the individual cells. You have to consider the overall structure, the function, because that imposes constraints on how those individual cells behave.
Constraints from the top down, influencing what happens at the bottom. That sounds a lot like the concept of downward causation.
It is. And systems biology offers concrete examples of how that might work, like the environment of a cell can influence the activity of its enzymes, or the physical properties of a tissue can constrain the behavior of the cells within it. So downward causation isn't just some abstract philosophical idea. It has real, measurable effects in biology. And of course, this all ties into the whole debate about emergent properties. Are they just a result of our limited ability to predict complex systems, or do they represent genuinely new properties that emerge at the system level?
So what about predictability? Systems biology relies heavily on computational models, simulations. Does that mean eventually we'll be able to predict everything if our models just get good enough?
Well, there are different schools of thought on that. Some people are optimistic. They think as our models get more detailed, incorporate more interactions, our predictive power will skyrocket. Others argue that biological systems are inherently complex, nonlinear. There might be fundamental limits to how much we can predict, no matter how powerful our computers get. And then there's this tension with Occam's razor, right? The idea that simpler explanations are better. At what point does adding more complexity to a model actually make it less useful?
Okay, last but not least, how does all this affect our understanding of scientific explanation in biology?
Systems biology, with its focus on math, on networks, it really expands our view of what counts as a good explanation. Mechanistic explanations, the step-by-step causal accounts, those are still important. But systems biology also highlights other types of explanation.
Like what?
Well, there are topological explanations. Those focus on how the structure of a network, the connections, how that explains its behavior, almost regardless of the specific molecules involved. There are also design explanations, which explain why a feature exists by pointing to its functional advantage. And these can be seen as constraint-based explanations, emphasizing how physical and formal constraints shape what's even possible. This leads to the idea of explanatory pluralism, the view that different types of explanation might be necessary for understanding different aspects of biological systems. And some people are even trying to come up with ways to integrate these different types of explanations into more comprehensive frameworks.
So it sounds like systems and synthetic biology, they're not just giving us new answers, they're making us rethink some of our basic assumptions about how we explain the world, about life, about knowledge itself.
I think that's a great way to put it. And it's not just about explaining, right? It's about predicting, controlling, designing new biological capabilities, figuring out the minimal requirements for life. All of this has huge philosophical implications.
Well, this has been a seriously thought-provoking deep dive. For anyone listening who, loves a good philosophical puzzle, it's clear that these fields are a goldmine. They're giving us new ways to think about some of humanity's oldest questions.
Absolutely. And thinking back to our discussion about, that thin notion of design, the role of network architectures, it raises a really interesting question. To what extent can we understand what a complex system can do, whether it's biological or not, just by looking at its structure? Can we ignore the specific materials, the history that led to that structure? It's a really fundamental question about the relationship between form and function.
A lot to chew on there. And if this has sparked your curiosity, I'd encourage you to explore some of the philosophical literature on reductionism, emergence, all that. There's also a ton of fascinating work being done on the ethics of synthetic biology, the metaphysical implications, all that. It's a really exciting area where biology and philosophy intersect. Thanks for joining us on this deep dive.